What I'm about to tell you shouldn't exist according to every law of physics we know, but I've seen it with my own eyes, measured it with my own instruments, and it's changed everything I thought I understood about life on Earth. As a marine thermobiologist who's spent 8 years studying how organisms survive in extreme temperatures, I thought I'd seen every impossible adaptation nature could create. I was wrong. In the Arctic waters north of Svalbard, I discovered fish that generate internal temperatures exceeding 200 degrees Fahrenheit while living in water that's below freezing. And what we learned about how they do it will terrify and amaze you. My name is Dr. Elena Vasquez, and I've dedicated my career to understanding how life persists in the most hostile thermal environments on our planet. After completing my doctorate at Woods Hole Oceanographic Institution, I've spent 5 years conducting research in Arctic waters, discovering 17 new species of cold-adapted fish, and publishing over 30 papers on extremophile biology. I've studied organisms in hydrothermal vents, analyzed bacterial life in Antarctic ice sheets, and documented fish survival in sub-zero temperatures that should freeze their blood solid. Every discovery has followed predictable patterns based on known physics and biochemistry. Antifreeze proteins, metabolic adjustments, cellular membrane modifications, all explainable, all within the boundaries of what science considers possible. But nothing in my training prepared me for what we encountered during our expedition to map thermal variations in Arctic fish populations. The mission began as routine research funded by the National Science Foundation to study how climate change affects thermal regulation in Arctic marine life. Our team consisted of myself as lead researcher, Dr. James Chen, a biochemist specializing in protein analysis, Dr. Sarah McKinnon, our marine ecosystem specialist, and Dr. Yuki Tanaka, our thermal dynamics expert. We were aboard the research vessel Polar Dawn, equipped with the most advanced thermal imaging equipment available, including infrared cameras capable of detecting temperature variations as small as 1/10 of a degree, underwater thermal probes with millimeter precision, and a collection system designed to maintain live specimens at their natural temperatures. Our goal was to document baseline thermal signatures of Arctic fish species before climate change altered their environment permanently. The journey to our research coordinates took 3 days through increasingly hostile waters. Pack ice formations grew thicker as we traveled north, requiring our ship to break through barriers that groaned and cracked around us like the planet itself was warning us away. The temperature dropped to -38 degrees Fahrenheit, with wind chills that made exposed skin freeze in under 2 minutes. Our target location was a deep water channel between 2 ice shelves, where previous research had documented unusual fish populations that seemed to thrive in conditions that should be lethal. Local Inuit guides had mentioned these waters in their traditional knowledge, describing fish that carried fire in their bellies and could melt ice with their presence. But we assumed these were metaphorical descriptions of normal cold water adaptations. For the first 48 hours, everything proceeded exactly as our research protocols predicted. We deployed our thermal monitoring equipment, established baseline water temperatures of -2 degrees Celsius, and began systematic collection of fish specimens. The initial catches yielded expected results: Arctic char with enhanced antifreeze proteins, polar cod with modified hemoglobin for oxygen transport in cold water, and several sculpin species with cellular adaptations we documented in previous expeditions. Every thermal reading fell within normal parameters. Every biochemical analysis showed predictable cold water modifications. Our instruments functioned perfectly. Our team worked efficiently, and I felt confident we'd complete another successful documentation of Arctic marine adaptation. But on the 3rd day, everything changed. Dr. Tanaka was conducting routine thermal scans when her equipment started displaying readings that made no sense. The underwater thermal probe, which had been functioning perfectly for 72 hours, suddenly began detecting temperature spikes that seemed impossible given our environment. Initial readings showed brief flashes of heat signatures reaching 60, then 80, then over 100 degrees Fahrenheit in water that was 2 degrees below freezing. We assumed equipment malfunction and ran diagnostic tests on every instrument. Hardware checks revealed no problems. Software calibrations remained accurate. control tests in known temperature water samples confirmed our equipment was functioning perfectly. Yet the anomalous readings continued, appearing randomly across different areas of our sampling zone, lasting anywhere from 30 seconds to several minutes, then disappearing completely. During the fourth day, the thermal anomalies became more frequent and more intense. We detected heat signatures reaching 150 degrees Fahrenheit, temperatures that should instantly kill any fish and create visible steam clouds in freezing water. Yet we observed no surface disturbances, no dead fish, no environmental changes that would indicate such extreme heat sources. Dr. Chen suggested underwater volcanic activity, but our geological surveys had detected no geothermal vents, no sulfur signatures, no mineral compositions indicating volcanic presence. Dr. McKinnon hypothesized equipment interference from magnetic fields, but our electromagnetic readings remained stable. As the team lead, I faced a choice between abandoning our research site or acknowledging that we were detecting something that violated basic thermodynamic principles. Then we saw them. Dr. McKinnon spotted the first specimen during our 5th day of monitoring, a fish approximately 12 inches long that appeared normal except for one impossible detail. The water around it shimmered with heat distortion, exactly like air above hot pavement in summer. Through our underwater cameras, we watched this fish swimming comfortably in -2-degree water, while generating enough heat to create visible thermal distortion extending 3 feet in all directions. Our thermal imaging confirmed what our eyes couldn't quite believe. This single fish was maintaining an internal temperature of 196 degrees Fahrenheit while showing no signs of distress, no tissue damage, no behavioral abnormalities that would indicate thermal stress. We attempted to capture this first specimen using our standard collection nets, but the moment our equipment approached within 2 feet of the fish, the metal components became too hot to handle, even through insulated gloves. The fish seemed aware of our presence, but showed no fear, no flight response, no defensive behaviors typical of deep-water species. Instead, it appeared curious about our equipment, swimming closer to our cameras and probes as if investigating our technology, with the same fascination we felt studying it. After 6 failed capture attempts, we realized our standard collection methods were inadequate for organisms that could generate enough heat to damage our instruments. Dr. Chen developed a modified capture protocol using ceramic-lined containers and thermal-resistant materials borrowed from our ship's engine room. Working in shifts to prevent hypothermia, our team spent 18 hours constructing a collection system that could withstand extreme temperatures while maintaining the sub-zero water conditions these fish apparently required. The engineering challenge seemed absurd, creating equipment to capture creatures that needed to be simultaneously frozen and superheated, violating every principle of thermal equilibrium we understood. Our first successful capture occurred on day 7, when we managed to secure a single specimen in our modified containment system. The moment we brought it aboard, our portable thermal sensors confirmed readings that made my hands shake with excitement and terror. This fish maintained an internal core temperature of 203 degrees Fahrenheit, while swimming in water that measured exactly -2 degrees Celsius. The temperature differential was over 200 degrees, a thermal gradient that should be physically impossible to maintain in a living organism without massive energy expenditure that would require constant feeding. Yet this fish showed no unusual feeding behaviors, no enlarged digestive systems, no anatomical features that could explain such extreme thermogenesis. Blood samples taken through specially designed thermal-resistant needles revealed biochemistry that defied everything we knew about protein stability and cellular function. Proteins typically denature and become non-functional at temperatures above 140 degrees Fahrenheit. Yet this fish's blood contained fully functional enzymes operating at over 200 degrees. Dr. Chen's analysis showed protein structures unlike anything in biological literature, with molecular bonds that appeared to strengthen rather than weaken under extreme heat. The hemoglobin molecules transported oxygen efficiently despite temperatures that should have broken down their structure completely. Cellular analysis revealed even more impossible characteristics. Mitochondria, the powerhouses of cells that typically function within narrow temperature ranges, were operating at peak efficiency while experiencing thermal conditions that should have destroyed their membranes instantly. Cell walls showed no signs of heat damage, no protein denaturation, no DNA breakdown that would indicate thermal stress. Instead, these cells appeared to thrive under conditions that would instantly kill any known life form on Earth. The fish's metabolism operated normally. Its organs functioned perfectly. Its nervous system responded to stimuli appropriately, all while maintaining temperatures hot enough to brew coffee. The breakthrough came when Dr. Tanaka discovered how these fish managed their impossible thermal regulation. Using microthermal probes, we mapped the temperature distribution throughout the fish's body and found that different organs operated at precisely controlled temperatures ranging from the surrounding water temperature of -2 degrees in their extremities to over 200 degrees in their core organs. The fish wasn't simply generating heat uniformly, it was maintaining multiple thermal zones within a single organism, with temperature gradients that shifted and flowed like a living thermal map. Even more remarkably, we discovered that these fish could control their heat generation consciously. When we introduced mild stressors like bright lights or loud sounds, the fish would increase their thermal output, raising their core temperature to over 220 degrees, while keeping their skin temperature exactly matched to the surrounding water. When relaxed, they could lower their thermal signature to nearly undetectable levels, while maintaining basic biological functions. This level of thermal control suggested not just biological adaptation, but conscious manipulation of physical processes we didn't understand. Further investigation revealed that these fish existed in small schools of 12 to 15 individuals that appeared to coordinate their thermal output collectively. When one fish increased its temperature, others in the school would adjust their heat generation to maintain optimal thermal distribution throughout the group. They were functioning as a collective thermal regulation system, sharing heat load and creating microenvironments that defied the freezing Arctic waters around them. The implications were staggering. These organisms had evolved not just individual thermal control, but collective thermal intelligence. Our research timeline was running short due to approaching weather systems, but we managed to collect tissue samples for genetic analysis. Dr. Chen's preliminary DNA sequencing revealed gene sequences that had no matches in any biological database. These fish possessed genetic material that appeared to code for proteins and cellular structures that didn't exist in any known life form. The genetic markers suggested an evolutionary timeline that didn't match any known Arctic species, as if these fish had evolved separately from all other life on Earth, or represented a form of life that operated under different physical laws. On our final day at the research site, we made the most shocking discovery of all. While reviewing footage from our underwater cameras, Dr. McKinnon noticed that the heat signatures from these fish were creating small currents in the surrounding water that appeared to prevent ice formation in a zone extending nearly 50 feet from their location. These fish weren't just surviving in Arctic waters. They were actively modifying their environment, creating thermal pockets that allowed other cold-sensitive organisms to survive in areas that should be completely frozen. They were functioning as biological heating systems for entire underwater ecosystems. The journey back to port took 4 days, during which we maintained our specimens in specially constructed thermal-resistant aquariums that had to simultaneously freeze and heat the water to keep these impossible fish alive. Every measurement we took confirmed our initial findings. These fish violated fundamental laws of thermodynamics while thriving in conditions that should be lethal. Our data was so far outside normal parameters that we knew no peer review process would accept it without extensive verification from multiple independent sources. Upon returning to civilization, we faced the challenge of presenting impossible findings to the scientific community. Our first attempts to publish were rejected by 3 major journals, not because our methodology was flawed, but because our results were deemed thermodynamically impossible by reviewers who refused to consider that our measurements might be accurate. We spent 6 months conducting additional verification tests, bringing in independent thermal measurement specialists, and documenting every aspect of our findings with redundant instrumentation. The breakthrough in acceptance came when Dr. Tanaka presented our findings to a conference on exotic matter physics. Theoretical physicists who studied quantum thermodynamics found our data fascinating rather than impossible, suggesting that these fish might represent the first documented case of biological quantum thermal regulation, where organisms operate using principles from quantum mechanics that allow them to transcend classical thermodynamic limitations. The possibility that life could evolve to manipulate quantum properties for survival opened entirely new fields of research. Follow-up expeditions by other research teams have confirmed our findings and discovered additional populations of thermophilic ice fish in Arctic waters previously thought to be biologically dead due to extreme cold. These fish appear to be expanding their range as Arctic waters become more accessible due to climate change, suggesting they may play a crucial role in future Arctic ecosystems. Some researchers hypothesize that these organisms could be harbingers of life forms that will dominate as Earth's climate becomes more extreme. The implications of our discovery extend far beyond marine biology. If life can evolve to transcend basic thermodynamic principles, it suggests possibilities for biological thermal regulation that could revolutionize everything from medicine to engineering. Understanding how these fish generate and control extreme heat while surviving in freezing conditions could lead to breakthroughs in thermal management technology, medical treatments for thermal injuries, and even space exploration technologies for surviving extreme temperature environments. What started as routine research to document climate change impacts on Arctic fish became a discovery that challenges our fundamental understanding of what life can achieve under impossible conditions. These thermophilic ice fish prove that nature continues to surprise us with solutions to survival challenges that exceed our scientific imagination. Every measurement we took, every test we conducted, every analysis we completed confirmed that life on Earth is far stranger and more resilient than we ever believed possible. The research continues, with new expeditions planned to study how these remarkable fish evolved their impossible thermal abilities and what their presence means for the future of Arctic ecosystems. As climate change reshapes our planet's most extreme environments, discoveries like the thermophilic ice fish remind us that life finds ways to thrive under conditions we consider impossible, and that nature's solutions to survival challenges often transcend the boundaries of human understanding. If this discovery amazed you as much as it changed my understanding of life on Earth, smash that like button and subscribe for more scientific discoveries that challenge everything we thought we knew about the natural world.